right, Chapter 8. Chapter 8 is going to be about cellular respiration. In this chapter, we're only going to be covering two sections. Number 1, the overview of cellular respiration, and then 3, we're going to talk just a little bit about fermentation. So we're going to start off with cellular respiration. It is the process by which cells acquire energy by breaking down nutrient molecules that have been produced by the photosynthesizers. Now, unlike photosynthesis, cellular respiration is going to use up oxygen and produce carbon dioxide. This involves the complete breakdown of glucose to carbon dioxide and water. Glucose is a high energy molecule, while carbon dioxide and water are low energy molecules. So, going back, does this mean that energy will be absorbed or released by this reaction? Is this an endergonic or an exergonic reaction? I'm going to leave that up to you to decide. But ultimately, energy is going to be released a little bit at a time in a stepwise fashion, which allows for the efficient production of ATP from these reactions. ATP will slowly and gradually be produced at each step. If it were to all occur at once, most of the energy would be lost as heat. So we're going to release just a little bit of ATP at a time. We're ultimately going to be using NAD plus and FAD as coenzymes for these reactions. So here's just kind of an overview of cellular respiration. When an ocelot breathes, it's going to acquire oxygen from the air. And when it feeds on a lizard, it's going to acquire glucose. So that's what we have here. We have an ocelot eating a lizard. So he gets his nutrients from his food. He gets oxygen from the air. Both molecules are going to enter the bloodstream and then are going to be carried to the body cells. Once inside the cell, we're going to diffuse over to the mitochondria, where cellular respiration is going to occur. Carbon dioxide and water are going to be released as glucose breaks down in the mitochondria to provide the energy for ATP production. So here's just a really great summary slide that shows oxygen comes in, glucose comes in, carbon dioxide and water come out, and we are ultimately producing large amounts of ATP. Cellular respiration is going to involve multiple metabolic reactions, and each reaction is going to be catalyzed by a unique enzyme. One type of enzyme that we're going to pay attention to will be the enzymes that use the coenzyme NAD to speed up their reactions. NAD is going to become oxidized. This is where it has to give up an electron. So what sort of charge will an NAD molecule have if it gives up an electron? It's going to be positively charged. That's why we draw it as NAD+. It's going to have a positive charge because it has given up an electron, so now it has one fewer electrons. Electrons are negative charges. If we give up a negative charge, we become a positively charged molecule. When NAD+ accepts an electron, we say that it has been reduced. Reduced means that it has gained an electron. The acceptance of electron usually coincides with the binding of a hydrogen molecule. So NAD+ will become NADH. FAD, flavin adenine dinucleotide, is also a coenzyme of oxidation reduction. Sometimes it's used instead of NAD+. It's going to accept 2 electrons and 2 hydrogen atoms to become FADH2. So we're going to talk just a little bit about the different phases of cellular respiration. Cellular respiration is going to occur in 4 separate phases: glycolysis, the prep reaction, citric acid cycle, and the electron transport chain. We're going to start off by going through glycolysis. Glycolysis, from the Greek glyco meaning sugar and lysis to split apart. It's going to be the breakdown of sugar into, or the breakdown of glucose, which is a 6-carbon molecule, into 2 3-carbon molecules called pyruvate. This process is going to result in the production of NADH and the net gain of 2 ATP molecules, and is ultimately going to occur in the cytoplasm. Now here's the interesting thing. Glycolysis does not utilize oxygen. At this point, we're only in the cytoplasm and no oxygen has been required for this step. One thing I would like you to know for each of the phases of glycolysis, I would like you to know generally what goes in, what comes out, and where it occurs, and whether or not it uses oxygen. So location, what goes in, what comes out, and whether or not oxygen is required. If a system does not require oxygen, we say that it is anaerobic. An meaning without. Aerobic reference to oxygen. So it does not require oxygen for an anaerobic reaction. An aerobic reaction, so if you were to get rid of that an and just be aerobic, reaction does require the input of oxygen. So the next step is going to be the prep reaction. The prep reaction occurs in the matrix of the mitochondria. Those two pyruvate molecules are going to be broken down from 3 carbon to 2 carbon molecules, and a single molecule of CO2 is going to be released from each one. So we're going to oxidize that pyruvate, and they're going to enter the mitochondria. The electron energy is going to be stored temporarily as NADH, and 2 carbons are going to be released as carbon dioxide. Again, this does not require oxygen. This reaction is simply prepping the materials for our next step. The next step is going to be the citric acid cycle. The citric acid cycle also occurs in the matrix of the mitochondria. Each of the 2 carbon acetyl groups produced by the prep reaction are going to match up with a 4 carbon molecule forming 2 6 carbon citrate molecules. And the breaking of those citrate bonds results in the production of NADH and FADH2. 4 carbon dioxide molecules are going to be released as a result, 2 from each citrate molecule. And it's going to produce 2 immediate ATP molecules per glucose molecule.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
So the electron transport chain is next. At the electron transport chain, this is going to be a series of carriers on the cristae of the mitochondria, as we talked about in our previous chapter. This is where we're going to extract the energy from NADH and the FADH2. And the way we extract energy is by pulling off those hydrogen ions. We pull off those hydrogens from NADH and FADH2. As we pull them off, we're going to put them across the plasma, or put them across the membrane of pull them across the cristae, the membrane of the mitochondria. As we do this, we're going to be passing electrons from higher to lower energy states. When we do this, we're going to create a very high concentration gradient of proton ions inside of the matrix. As those hydrogen ions flow through the matrix out into the rest of the mitochondria, we're going to pass through an ATP synthase pump, which is going to produce ATP as hydrogen ions pass through. This step produces 32 or 34 molecules of ATP by chemiosmosis. This is our hugely efficient step in producing ATP.
When glucose is oxidized during glycolysis and the citric acid cycle, NAD+ and FAD are reduced to NADH and FADH2, and hydrogen ions, H+, are released. In the mitochondria, the electrons from NADH are transferred to the electron transport proteins, and the hydrogen ions are transferred across the membrane. As the electrons move from carrier to carrier down the electron transport chain, more hydrogen ions are carried across the membrane. The terminal electron acceptor for this process is oxygen. When oxygen is reduced, water is produced. The transfer of hydrogen ions generates a potential energy gradient across the membrane of the mitochondrion. Because membranes are impermeable to ions, the hydrogen ions that re-enter the matrix must pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate by the ATP synthase complex.
Next up, we have a summary slide that goes through all of the different phases of glucose breakdown. And I've got a little summary video that goes along with it.
The energy within a glucose molecule is stored in its chemical bonds. Cellular respiration is a four-stage process which releases this energy and forms ATP. These reactions occur both in the cytoplasm and mitochondria of the cell. The first of these reactions is called glycolysis, and it occurs in the cytoplasm of the cell. A series of chemical reactions breaks down glucose to pyruvate, in the process generating a net of 2 ATP molecules. In addition, a small amount of NADH is generated, which will be used later in the electron transport chain. As the pyruvate molecules move into the mitochondria, they enter the preparatory reactions. These reactions rearrange the chemical bonds of the pyruvate, forming acetyl-CoA. In the process, some NADH is released. Following the preparatory reactions, the acetyl-CoA molecules enter the citric acid cycle, also referred to as the Krebs cycle. Here, the remaining chemical bonds are broken, generating 2 molecules of ATP, carbon dioxide, and several molecules of NADH and FADH2. The final set of reactions are collectively called the electron transport chain. The inputs to the electron transport chain are the NADH and FADH2 molecules from glycolysis, the preparatory reactions, and the citric acid cycle. This stage of cellular respiration uses chemiosmosis to produce the majority of the ATP needed by the cell. It is here that oxygen is used as an electron acceptor, producing small amounts of water. For each glucose entering these pathways, between 36 and 38 ATP molecules are produced.
All right, and so that's sort of a summary of the 4 phases of complete glucose breakdown. You should be able to talk through this diagram and be comfortable with explaining what is happening at each step of glycolysis, prep reaction, citric acid cycle, and the electron transport chain.